curious, serious tone. Hematological cancers, like leukemia, are lethal because they have evolved a devastating trick. They turn themselves invisible. To a patient's natural T cells, the microscopic soldiers meant to patrol the bloodstream, these malignant cancer cells simply do not register as a threat. The biological solution is surprisingly straightforward in theory. If we can equip those blind T cells with a specific tracking system, they will recognize the cancer and destroy it. But the human body has no internal mechanism for upgrading its own cells. To bridge that gap, scientists developed a radical procedure. They extract a patient's white blood cells, transport them to a laboratory, rewrite their genetic code to physically install the tracking system, and then infuse them back into the patient's bloodstream. Most conventional therapies rely on static chemical compounds stamped out by the millions. Because this process engineers a bespoke, living treatment out of the patient's own biological material, it creates a massive logistical challenge, requiring a flawless sequence of specific engineering solutions for every single person. The physical core of this upgrade is a synthetic blueprint called a chimeric antigen receptor, or CAR. This diagram illustrates how the CAR operates. Top, the ectodomain acts as an external sensor, physically binding to specific cancer antigens. Below, the transmembrane domain anchors this hardware into the cell. Finally, the intracellular signaling domain translates that connection into a chemical command to attack. We have the perfect microscopic weapon completely mapped out. But a molecular blueprint has no clinical value unless scientists can figure out how to physically install it into millions of living cells at once. That brings up an immediate mechanical problem. You cannot simply force a new genetic blueprint into a delicate T cell without destroying it in the process. To bypass the cell's natural defenses, scientists hijack nature's most efficient infiltration tools, lentiviral vectors. These are highly engineered derivatives of the HIV-1 virus, chosen specifically for their unique ability to slip smoothly across an intact nuclear envelope and stably integrate new DNA directly into the host genome. Injecting an HIV-derived virus into a compromised patient sounds alarming, so bioengineers rely on a third-generation, self-inactivating vector design. They secure the virus by stripping out regulatory genes, splitting the remaining code across 4 separate plasmids. By assembling from fragments lacking replication instructions, the virus retains its ability to deliver the CAR blueprint, but has zero capacity to multiply. This process yields a completely harmless, highly efficient genetic delivery vehicle. But to treat even a single patient, manufacturers have to mass-produce millions of these precise microscopic vehicles. To meet that demand, the process shifts from the laboratory bench to industrial scale in a phase called upstream bioprocessing. Scientists move the operation out of small flat plastic flasks and into massive single-use stirred tank suspension bioreactors. Inside these towering tanks, specialized HEK293T producer cells float freely in a precise chemical broth. Because they don't require highly variable animal serum to survive, these cells can safely divide to incredibly high densities, acting as microscopic factories churning out raw viral vectors. Once the bioreactors hit peak capacity, the process flips to downstream bioprocessing. The pure viral vectors are now floating in a murky broth of cellular debris, waste proteins, and raw DNA that must be completely removed. The harvest undergoes a rigorous mechanical gauntlet. First, high-speed centrifugation separates heavy debris. Then, liquid passes through depth filters before hitting ion exchange chromatography, which separates out the remaining microscopic impurities based entirely on their electrical charge. The result is a highly concentrated, biologically pure vial of the viral vector. But the delivery vehicle is only one half of the equation. The other half involves securing the fragile, living T cells directly from the patient. The clinical phase begins with leukapheresis. At the hospital, the patient's blood is pumped into a continuous flow centrifuge, which carefully skims off the required white blood cells, the leukocytes, and safely returns the remaining plasma and red blood cells back into the patient's arm. These extracted cells are highly sensitive to temperature and time. To survive the complex transit from the clinical collection center to the centralized manufacturing facility, the cells are immediately plunged into deep cryopreservation. Upon arrival at the manufacturing hub, the frozen cells must be carefully revived. Facilities utilize specialized dry thaw systems, where warmed cushions press against the bags to strictly prevent any waterborne contaminants from coming near the material. This triggers the final convergence. The thawed patient cells and the purified viral vectors are injected into a completely sealed, automated robotic system, like the Clinimax Prodigy. Inside the machine's sterile tubing, the virus infects the targeted T cells, permanently installing the CAR blueprint and triggering the cells to multiply by the millions. By executing the selection, infection, and expansion phases entirely within a single closed robotic loop, manufacturers remove human error and ensure the final drug product remains absolutely sterile. The newly minted, fully armed CAR T cells undergo one final round of cryopreservation. They're shipped back across the country to the clinic, thawed, and infused directly into the patient's bloodstream. 3 decades ago, immunotherapy was restricted to a fragile series of isolated, slow-moving experiments confined to individual laboratory hoods. Today, that science operates at an extraordinary industrial scale. Highly regulated CAR T therapies are FDA-approved, mass-manufactured, and actively deployed to combat severe leukemias and lymphomas across diverse patient populations globally. The biological payoff of engineering a living therapy is its persistence. Because the engineered cells remain alive inside the body, they continue to patrol, identify, and destroy targeted cancer cells, yielding remissions that clinical data shows can last for a decade or more. Delivering these decade-long remissions at scale is no longer a matter of bench-side theory. It is a test of the industrial bioprocessing and logistical precision required to successfully manufacture living, patient-specific therapeutics for every